there's a lot of choices that have to be made in that pre-processing pipeline, and usually when people have analyzed their data, they've made different decisions about the statistical measure to use for time series correlation in an fMRI or the tractography algorithm. So what we're sort of doing is leaving that to the researcher to decide which metric they want to use to define connectivity, and then we come in to at the matrix stage. Okay, so this is the home page for our UMCD site that you land on, and right away what you can do is you can analyze any network that's been publicly shared. So I'm going to log out here first, and what I'll do is if we go here, so there's anytime someone uploads the data, they choose a study name, and so these are examples of data that me and a colleague have uploaded. Not a lot of data has been shared on here yet, but so this was some data from the ICBM data set, and so we analyzed some diffusion tensor imaging from one subject and calculated the whole brain structural connectivity matrix from that subject. So, so when I choose the ICBM study here, all of the matrices for that study that have been shared show up on this list here. So I'm going to choose this, this 1004 DTI file. For this analysis, one thing I didn't mention on the when I was talking about graph theory is that one decision that has to be made is whether to leave the matrix that you have in a weighted form such that the different connections have different strengths or weights. The alternative method is to binarize the network, which sets all connection strengths that exist to 1 and everything else to 0. These two methods have their pluses. I'll go with a weighted network analysis here, and I'm going to choose to include 20% of connections that are in the network. So this is just going to keep, I believe that this matrix is about 25% by nature. By setting this to 20%, what I'm doing is excluding the 5% of connections that are weakest in the network. You can choose the orientation for the output images. I'm going to stick with axial for the moment.
Just a second. You chose 20% of the edges.
Yes.
And from a weighted graph, you did not say whether you are choosing the strongest 20% or weakest 20% or any other way you characterize 20%.
Right. Sorry about that. So typically when you do folding step, you keep the options. There's no option that those carry the most information, the most interesting. So there's a few assumptions that we make on the site when we analyze the data, and one is that you want to keep the strongest connections. And so those sort of underlying decisions that we make, we try and sort of disclose all of that on the page that I'm highlighting.
Okay. All right.
Here.
Yeah. Okay. So these.
Okay.
All right.
So, so yeah, what you see here first is the information on the network. So this is information that the researcher in when they uploaded the data. In a bit, I'll get to how you can upload your own data to the site here. So the name of this network is you can see. The atlas was the free surfer atlas, so the free surfer cortical and subcortical parcellation was performed on this subject, and that was the set of regions that were used. The data is DTI. This was a normal subject. The group size means that this was an individual rather than an average across several subjects. The age range was just 45 because that's the age of the subject, and then there's also some pre-processing notes on how we pre-process the data in order to derive the matrix. So in this case, you can see that the raw density of the matrix before we applied a threshold was 27%. Our chosen threshold was 20%. And then these are global measures that I was talking about earlier, the characteristic path length, in the clustering coefficient. These numbers, especially for a weighted network analysis, are really only useful to interpret in the context of comparison to an equivalent set of subjects. They are proportional to the edge strength in a network, which may have undergone some normalization or some transformation. So the values here are reflective of underlying weights in the network. I saw there's a question.
Yeah, just this, Jonathan. I just have a question. So, what's the difference between the raw? We chose 20% as the density, but what is the raw density percentage a measure of?
So when I uploaded this matrix, it was I uploaded the raw matrix, and that's what we're trying to get people to upload, is something that hasn't been pre-threaded. It's just the raw connection strengths that they've detected. And the matrix that I uploaded, only 27 of the regions connected to each other. The remaining 70% had no connection strength between.
Okay.
Does that make sense?
Yeah.
Okay. Great.
It also looks, just I'm going to really mess up your name, but Queen Yang Li know that DTI is about 50 megabytes.
50 megabytes.
And then it's JAM. So.
Oh, okay. So yeah, maybe 50 times bigger than the matrix you derived, based roughly on the regions you have in that. Okay. The number of components in the network is an indication that this network is entirely connected, that there's no subsets of nodes that have no other connection to the rest of the network. If you apply a lower threshold, like 10% connections or 5% of connections, usually what happens is you see the network fragment into these different components. So you can see here that at this threshold, the network is totally all interconnected. These small attributes I didn't talk about too much, but what small worldness looks at is a show of the clustering coefficient, the average clustering coefficient in the network, to the path length. And it's biological networks tend to have this sort of small world configuration. Some of you may be familiar with this. For those of you who aren't, the idea is that the regions tend to form local clusters with these, but there are a few long
Disable that phone number for next webinar, but apparently
someone accidentally left the phone on.
Sounds like someone's being eaten by a large dragon.
Yeah, there might be some eating.
I was thinking, I mean,
They may not be able to mute. Okay.
Yeah, they may not be able to mute.
Bigger problem.
Yeah. All right, thank you.
Okay.
I think we'll just...
Try. We'll trudge on here. Okay, so I won't go too much into these now. I try and some of the best references for more detail on each of these measures on the About page. First, I'll show you this 3D representation. So this is using WebGL to get a 3D representation of the network. And so what we can see is the regions that are connected. So it's Java. And I just made a few modifications to that script to be able to plot these matrices in 3 dimensions like this. I saw there was an highlight about this image is that the radius of the spheres is corresponded to different measures. So in this case, the radius of each sphere is corresponded to the degree of that um, which is just how many connections are coming into that node. So regions that have more connections have a larger degree. And there's different measures like the clustering. If you click here, and like any good demo, it seems like it's having a problem. Try and show you the modularity here, and it looks like it's computing right now. Okay, so in this case, the radius of each sphere is 1, and you can see these different modules highlighted in these different colors. So this is one feature here. I see I'm taking my time here, so I'm going to try and coming into it or out of it. And what you can see is that most in this network have a degree of about 16. Highest and the highest nodes have about a degree of 18. So these degree histograms can contain some interesting information about the about how large certain hubs can be in the brain. And then these images are 2D representations of the same thing we're looking at. So this is an axial or a top view, you could say. 2D representation of the modularity. What you can see in this network is that the modules tend to align here. So there's this, you could maybe say visual network, and then these more sort of, and then moving forward. The image on the right is what's called a force embedded or spring force diagram. It's the same model regions from the image on the left, and you can see that the abbreviations for the regions are the same, but it's decomposed it, or re-represented it in two dimensions, where the distance between any node in this image is an indication of how near they are to each other in the network, or how near their modules are. And you can see that the red and blue, light blue nodes are closer to each other in this in force embedded space than they are in physical space. And the interpretation of these can be tricky, but it's an interesting way to sort of abstract the connection diagram in order to some spatially distant nodes that may be more closely connected in this space. So I'm going to try and speed up here, go to this, to networks here. I'm going to log in. One feature that I mentioned is that when you share data, you have the option to make the data private or public. If it's public, then it's obviously viewable to anyone. If it's private, it's only viewable to you once you log in. So it's a way to, if you want to compare your data to data that's already been publicly shared, then you can put it up here and it's private and no one has access to it but you. So and the site's very secure, so it's a safe thing to do if you choose to use it. So I'm going to compare a couple networks from some DTI data here. Oops. Okay, I'm going to run a binary analysis, or actually I'll make this weighted. And again, I'll go with 20% sparsity, and then if I click Analyze here, this may take a little while to load, but what it's going to show us is essentially the same report from that I was previously demonstrating, but it's going to show it side by side for the two networks, so you can compare the measures here. So we see the network information for both of the networks, and then we can some of these measures.
Haven't you chosen the exactly same network?
Yeah, you're right. I have. Let me fix that. Okay, so this is from people who have the APO4 allele or the APO3 allele, and looking at their structural DTI networks. And we're working to get a loading bar here, so you have an indication of how soon it loads. Complete these analyses for networks that have 200 nodes happen in usually less than 30 seconds. And so if we look at this here and look at the global measures, we can see that this APO4 group has a higher clustering coefficient and also a higher path length than the APO3 carriers. So a more densely connected network, or path length is indicative of lower efficiency or path for information to travel. Reflected in the small worldness is slightly greater for this APO4 network than it is for this APO3. So you can see some differences. Modularity values are pretty similar. obviously we've just got two networks here, so we can't do statistics on that, but we can get some, qualitative information about the similarity or difference of networks. You can see that matrices are similar, and these images, you can't immediately look at them and see differences in the networks a lot of time. But if you look at like the between the centrality, we can see some interesting differences where for the APO3 group, it looks like this occipital fusiform area has higher between the centrality, or in the APO4 carriers, it looks like the putamen has greater between the centrality than the APO3 carriers. And if you go to the regional report for these, then you can get those specific measures for, so the APO3s are on top and the APO4 group is on bottom, and you can compare the equivalent measures in this way. And then one of the most interesting things about this group is a difference in modules, where you see on the left the APO3 carriers have this one consistent module that spans the entire midline of the brain, whereas in the APO4 carriers we see it segmented into a portion that starts at the posterior cingulate and goes to the frontal cortex, and also this posterior portion that may be connecting the precuneus to the hippocampus, or there may be, it may be that this connection has fragmented in the E4 carriers and may have, implications for information transfer in this network. So this is one of the applications of this. So in terms of uploading data to the network, so here's the upload form, and I had some data prepared to you how to upload, but I seem to have misplaced it. But basically there's a form where you would name your study, and so the general name for the entire study, and then you would typically give a unique identifier to each matrix for that study that you're going to upload. So so you're the owner of that study name, and then you would assign network names so that you can find them, and also so that others who are looking at your know how to choose individuals from that. You also
Very quickly though, is there any way that we could, for example, at NIF, grab those names and register them, or at least grab the URI of those data sets?
Yeah, so let me go to the browse data page here. So this is showing all of the data that's either publicly shared or that I have privately uploaded. And so for example, like this study, the study name is UCLA ASD, and then the network name is the mean ASD fiber density 48. And so if I were to go to this view, download, edit, delete link and go here, okay, right, so yeah, currently we just use this generic ID. What we're planning to do is reconfigure this URL so that it has both the study name and the network name in the URL, so that if you had that information, you could link directly to this network. Does that answer your question?
Yeah, that answers my question.
Great. Okay.
The current way that we construct your excuse me, URLs to get back from this to each of these individual data points is I'm want to make sure that there's a robust URI-based way of doing that.
Sure. Yeah. So that's a change that I think I'm going to try and roll out as soon as possible.
We should also, through NIF, be able to grab it per study, so know something about the study. And so having consistent URIs between the two systems would make that a lot easier.
Right. Okay, so on this browse data page, you can see all the data that's been shared. In this case, I've shared all of this data, so I'm allowed to go to an individual study here, and then I can make changes to anything I want. If there's some information that I left out the first time, I can go there and I could make a change to the atlas or anything. You can also sort these. So if I wanted to see the studies by modality, I can click this link and you see all the DTI at the top, all the fMRI clustered here at the bottom. So this is a first pass way to isolate the data that you want. You could do it by age. The sorting function is currently not working exactly, but I'm going to fix that so that you could easily find studies from a specific age range. Right, and so this is also the place where you would come if you wanted to download the data for this specific. So I'm going to log out here, and then if I go back to that page and I wanted to get data from this study, then I'd go to View and Download, and then I can just click the file. So if I click the file for the connectivity matrix here, then I've downloaded this file, and then I've got it. So that if I want to run my own analysis wherever I like to do that, then I can get all of the information here. One thing I do need to cover is the files that are most important to upload when you're sharing data. So the first was that connectivity matrix that I just downloaded. The second is the name of each region in that matrix. In this case, I've got this file. So there were 68 nodes in this region that came from FreeSurfer, and these are the full name for each of those regions. And so those are used for the reports that I showed, where you see the different network measures for each region. And equivalently, there's a file that has the abbreviation for each node. And the abbreviations are important for generating those diagrams so that we have a small bit of text to show to label each in the 2D images. And finally, the XYZ center for each node. So in this case, it was just the center of mass for each region that was used. And so these are just space delimited, and these are MNI 152 coordinates. And we're really hoping that where possible, people can share data using the MNI coordinates so that they're defined in this common space, which will make performing meta-analyses a lot more it's a lot easier to do when the data has been transformed into the MNI space. There are certain cases where that may not be possible, but we're hoping that whenever we can get that, we will. Right, so that's the main information that's shared for a network. One thing that I don't think I really have time to show now is to model a lesioned network, where once you choose a network, you can click any of the nodes that you wish to exclude from that network, and then it'll show a comparison of the network with the lesion and without. On the About page here, I try and put all of the information about the descriptions of the different tools that we use, and some background on, for example, how you would derive the XYZ center for any of your regions, and then also some references and some links to the programs used to calculate the different network measures. Right. So I'm just going to jump back to the very end of the presentation here. It's all this. The whole site is Python-based. We use a web framework called Web2Py that has Python on the back end that we use to render all of the web pages. And one nice thing about this is that we have access to all of the libraries in Python. NetworkX is used for the graph theory analysis. Matplotlib is used to create all of the images, and then NumPy and SciPy are used for all types of scientific calculation like correlation and you name it. And then, like I mentioned before, we use WebGL and this ChemDoodle JavaScript library that's open source for the network visualization.
Jesse, I'm sorry, but it's about that time.
Okay.
And what I want to do is I want to thank you so much for sharing. Oh, and here's the next slide.
Yeah.
Well, and I do want to just say once again that if you have more questions, please address them to NIF, or if you'd like to stay for a little bit afterwards. But I would like to thank all of the different participants. People have been starting to drop off. So maybe we have time for one more question from the crowd.
Sure.
There seems to be a pause, so I think I'm gonna say that's a thank you from Wei-Lu.
Great.
Okay, so if anybody has any questions, that they didn't ask during, what?
This is Karen.
Hi.
Hi. Jesse, are you getting many contributions from people outside your group, or how is the information that's put in there curated, and if it doesn't comply with the description data as you've set it up?
Right. So in terms of contributions, haven't been a lot from outside yet. We have a few collaborators that have promised to upload data in the near future. A lot of people haven't publicly shared data yet. I think one way that we can speed up that process is I'm working towards sharing all of the connectivity matrices from the ADHD 200 public data and also from the 1,000 functional connectomes. And I think that once there's more publicly available data, that may create a little more momentum for people to start sharing more of their own data. And in terms of assessing the data that people are uploading, if it doesn't conform, there's not a ton of QC steps that we have on our end. If all of the files that we ask people to upload are text files, and so if they don't have the proper formatting, like, for example, as many lines on the regional names as there are nodes in your matrix, then it'll detect that and it'll reject your submission. So we do some first pass sort of thing, counting the number of regions, but we don't do a lot more in terms of sort of smart parsing of the data right now. We're mostly putting the onus on the researcher to try and get that right, and I have examples up on the site of how your data should be formatted and how it should look, so I'm hoping that people will use those examples to format their data right before it comes in.
Right. So does that new journal bring connectivity? Do they require contributors to the journal to deposit their information anywhere, much as a gene data would be required in a different journal? Or are people coming to you just because they know you, or do you see a paper published and you invite people, or how does that work?
So we're still working on that. I don't believe that brand connectivity has any submission requirements. I know they've tried to do that in the past with raw fMRI data and met some resistance when it was required. So it's hard to figure out where to do that, like compared to a protein crystal structure that you have to upload or something. So it would be great if we moved towards that. I think it just requires some more maybe standardization of this type of data. And what I'm hoping is that we're planning to get a publication on the site out in the next year, and that'll lift the level of awareness, and that then that we can get a little bit of a community and a mailing list, and then ask people to share connectivity matrix, have it, and it's really just, I think, about getting the momentum started on getting people to share their data here. So that's something that we're really going to focus on in the next year here.
Okay, thank you. I have what's probably a really naive science question as well.
Sure.
When you're talking about the hubs and the connectivity from the functional data, I mean, isn't that data really derived from the oxygenation and the microvasculature?
Typically, yeah. If you're running an fMRI scan and you have bold data, then yeah, it's about oxygenation and hemodynamics.
Right. So do you have, I mean, is that really what that's capturing, or do you have another exercise you can do here where you, just as you did with the DTI, you could actually map microvasculature and see how that relates to the DTI hubs and networks, or is that, in essence, really what you're doing anyway?
Well, so, and I don't know, I can't think of any studies that have looked at the relationship between um, functional connectivity between regions and the degree of microvasculature. Is that sort of what you're asking?
Yes. Mm-hmm.
Yeah, I don't think anyone has really looked on that. Most of these analyses have really been done with bold data, sort of with the assumption that the bold intensity value is an analog for increased neuronal metabolism, but that hasn't really been confirmed. But it would be interesting to do that analysis. People have compared the structural um, the structural networks to the functional networks and found some similarities, because I, I would argue that the structural networks are more intuitive to interpret because it's based on a physical connection that we're detecting as opposed to a
Right.
correlation in blood flow or oxygenation. And so people have, done correlations of the connection strengths and found that there is a relationship there. But when you actually do the graph theory analysis of a functional and a structural network side by side, they're very different. And so decomposing that difference into um, the different components, the oxygenation versus maybe time series correlation being the not being the best measure. There's a lot of things that are contributing to those differences, and that's